The aim of this presentation is to review what the original paper says about the connection between DNA methylation and the cause of type 2 diabetes and whether identified epigenetic changes translate into functional effects that affect the pancreatic B cell function. There are two types of diabetes, type 1 and type 2, both regulated by pancreatic islets. These play an important role in the development of type 2 diabetes. Environmental and behavioral factors are said to contribute to the cause of type 2 diabetes in addition to genetic predisposition. DNA methylation is involved in maintaining gene expression patterns associated with insulin resistance in type 2 diabetes. It occurs mostly in context of CpG dinucleotide as 5-methylcytosine. CpG are regions of DNA where cytosine nucleotide occurs next to a guanine nucleotide.
Samples used for this experiment were obtained from 16 cadavers of similar age, ethnicity, and BMI. It consists of 5 diabetics and 11 non-diabetic Caucasians. To perform DNA methylation profiling, the newly developed Infinium methylation assay was used. The assay detects cytosine methylation at CpG islands based on highly multiplexed genotyping of bisulfite converted genomic DNA, which are placed on a bead chip. 1 microgram of genomic DNAs were isolated from pancreatic islets and was treated with sodium bisulfite. Upon treatment with bisulfite, unmethylated cytosine bases are converted to uracil, while methylated cytosine bases remain unchanged. The bisulfite-treated DNA is subjected to whole genome amplification, which is increasing amount of limited DNA samples, useful for genetic disease research where DNA quantities are limited, but many analyses are required. The products are then enzymatically fragmented, purified from primers and enzymes, and then applied to the chip. The chip contains over 27,500 individual CpG sites spread across just under 14,500 genes. There are 2 bead types for each CpG site per locus. One of the bead types will correspond to the methylated cytosine locus, and the other will correspond to the unmethylated cytosine locus, which has been converted into uracil during the bisulfite treatment. The bisulfite converted amplified DNA products are denated into single strands and hybridized to the chip. Hybridization is followed by single base extension labeled with dideoxynucleotides, which is subsequently stained with a fluorescence reagent. The level of methylation for the interrogated locus can be determined by calculating the ratio of the fluorescent signals from the methylated versus the unmethylated sites. The Infinium methylation assay uses 2 different bead types to detect CpG methylation. The U bead type matches the unmethylated CpG site, while the MB type matches the methylated site. In the top figure, the unmethylated CpG target site matches the U probe, enabling single base extension and detection. It has single base mismatch to the M probe, which inhibits extension. If the CpG locus of interest is methylated, as shown in the bottom figure, the reverse occurs. This method quantifies methylation levels at specific loci within the genome.
A filtering was performed on the datasets for the CpG sites that showed significant differences in DNA methylation levels between the non-diabetic donors and the type 2 diabetic donors. As a result of this filtering, type 2 diabetes-related methylation changes were identified. The results obtained from this filtering are shown as a heat map, and that's the diagram labeled. The yellow bar on the left-hand side represents the control sample, the non-diabetic donors, whereas the blue bar on the right-hand side represents the type 2 diabetic donors. The green section shows a low percentage of methylation, whereas the red area shows a greater percentage of methylation. On the heat map, we can see that there are more areas than green in the control sample compared to the type 2 diabetic sample, which therefore indicates hypermethylation in the control sample and hypermethylation in the type 2 diabetic sample. A bootstrapping analysis was performed to further assess the results obtained in the heat map and to infer the degree of confidence that can be assigned to those results. A value of 0.85 was found. This further supports the results obtained in heat map and indicates significant support for the distribution between non-diabetic and type 2 diabetic samples based on the analysis of the CPGs contained in the filtered dataset.
The data demonstrated that human pancreatic islets undergo DNA methylation alterations in type 2 diabetes that are discernible by means of DNA methylation profiles. The outcome highlights two facts. One, diabetic DNA methylation profiles are more similar to each other than to the methylation profile of any control sample, indicating the possibility of a type 2 diabetes-specific DNA methylation profile. The second outcome highlights that DNA methylation changes are sufficiently pronounced to distinguish diabetic from control samples.